Hello, today I'll be sharing my work on linking morphometric patterns of the adolescent psychosis spectrum to different data modalities. Now, psychotic disorders like schizophrenia are increasingly viewed as clinical manifestations of phenotypes that exist on a continuum from health to illness. In line with this continuum, hallmark psychotic disorder symptoms also exist at subclinical levels in the general population, which can be described as psychosis spectrum or PS features. These are most common during youth, a period of dynamic maturation, and also increase the risk of threshold psychopathology in later life. So this gives impetus to understanding the earliest emergence of PS features, which my work focuses on. As the psychosis spectrum is multifaceted, an analytical approach that considers its neurodevelopmental, neurobiological, clinical-behavioral, and molecular dimensions is important. And to this end, magnetic resonance imaging provides the chance to capture a neurobiological signature of symptoms via indices of different cortical metrics, as shown here. These features mature on different timelines and are thought to reflect different cellular level processes, and thus contribute complementary information in the context of abnormal neurodevelopment. Notably, PS cohorts show changes in individual features that mirror those seen in psychosis patients. However, it remains unclear whether the PS is also linked to changes in the coordinated variation of multiple features. Interestingly, work on morphometric similarity mapping has shown that examining covariance of multiple structural features can reveal brain networks that capture greater biological similarity, such as in cytoarchitecture, than single feature-based covariance networks. And while this specific technique is limited by an inability to recover individual feature contributions, more broadly, integrating multiple metrics may refine our understanding of clinically relevant differences in brain structure. Moreover, most PS studies have related a single domain or summary score of symptoms to brain measures, which fails to capture the heterogeneity of PS expression. Indeed, growing evidence points to the PS as emerging from the simultaneous variation of distinct symptom dimensions, where each dimension describes a phenotypic axis underlying multiple measurable symptoms along which a person can vary. And while a consensus model for the PS is still lacking, particularly for youth, such data-driven symptom dimensions may better capture major axes of neural variation. And so to investigate how morphometric phenotypes in the PS might confer risk for symptom patterns, we seek to integrate insights from cognition-related functional activation maps, gradients of cortical organization, and gene expression of cell classes. First, we aim to link morphometric covariance patterns to psychosis spectrum symptom dimensions in youth. And here, using the PNC, which is a non-help seeking community sample of youth, we followed previous papers to derive a subsample enriched for PS features. We then analyzed structural MRI data along with PS symptom profiles and sociodemographic measures from these youth. And to characterize multivariate neuroanatomy, we subjected 4 cortical metrics of interest after removing the influence of global measures to orthonormal projective non-negative matrix factorization. This decomposed the metrics into spatial covariance patterns called components and subject level weights of metrics onto components, where a stability analysis determined the number of components. And using this technique allowed us to identify contributions of individual metrics, as well as obtain sparse and non-overlapping components. We also ran an exploratory factor analysis to estimate major dimensions of PS symptoms and computed subject scores onto each symptom factor. Then to relate subject-specific morphometric component weights to symptom factor scores and demographic features, we ran a behavioral partial least squares, which found maximally covariant patterns of the 2 data sets. Now, from the NMF analysis, we identified 8 morphometric components, and the subject metric weights show that components were not driven by any single metric. Neuroanatomically, they captured largely bilateral and contiguous patterns, including the lateral medial prefrontal cortex, as well as inferior and anterior middle temporal lobe. Meanwhile, our factor analysis revealed 3 psychosis spectrum symptom dimensions, which were negative and disorganized symptoms, disordered thoughts or sense of self, and hallucinations and abnormal perceptions. Finally, our behavioral PLS analysis revealed 2 significant latent variables, but we focused on LV2 as it captured a symptom morphometry relationship. Specifically, older age, male sex, African American race, and higher scores on the disordered thoughts or sense of self dimension covaried with cortical changes, including lower gyrification components 1 and 4, higher and lower cortical thickness in components 1 and 3, lower mean curvature in component 2, and lower surface area in component 6. And we'll refer to these as components linked to disordered thoughts or sense of self. To relate components to brain function, we first used Neurosynth to meta-analytically explore functions spatially associated with each component. And we observed that components correlated with distinct domains of psychological term maps, summarized here. And interestingly, components linked to disordered thought or sense of self were associated with several of the higher order processes, including executive control, emotion, and semantic memory. Next, given converging evidence for a large-scale unimodal to transmodal cortical gradient of functional organization, we sought to situate morphometric components along maturational versions of this gradient. We show here component gradient value distributions from the adolescent principal gradient, and among components linked to disordered thoughts or sense of self, we see that components 3 and 4, and 6 and 1 sat near the unimodal and transmodal ends of the gradient respectively, while component 2 occupied a more intermediate position. Integrating child and adult expressions of these gradients, we see that components linked to the same symptom dimension occupy more unimodal positions in childhood, such as 2 and 3, shift in position most dramatically along this gradient across maturation, while more heteromodal components such as 6 and 1 have stable relative positions, but still show visible changes in the distribution of gradient values. Finally, we asked whether specific cell types underlie the components we've suggested to be vulnerable in the PS. And here we use the Allen Human Brain Atlas to extract tissue samples corresponding to components and input sample gene expression data grouped by component to mean-centered PLS. And this method finds relationships between groupings in a single data set and produced latent variables describing gene expression patterns that maximally differentiated the input components. By thresholding the output gene weights, we obtained PLS positive and negative gene sets describing different components, and then in each set computed the ratio of genes preferentially expressed in each of 7 brain cell classes. So we focus here on the first significant LV, which explained most of the covariance. In this plot, components separable by the LV have opposite sign contrast values, and the magnitudes show how well components are described by the corresponding gene weight pattern, or in other words, their contribution to the contrast. So components 4 and 6 most strongly express the PLS positive gene pattern, while component 2 most strongly expresses the PLS negative gene pattern, meaning this LV primarily separates components 4 and 6 from component 2. Interestingly, we observed that all components with positive contrast values had been linked to disordered thoughts or sense of self. And to better characterize the PLS positive and negative genes, we looked at their cell type enrichments. Here, the circles denote the true enrichment of each cell type. The box plots are null enrichment ratios from random gene lists, and the direction of difference between the two indicates whether a cell type is over or under enriched in the gene set. And we observe that PLS positive genes, which are more expressed in the red components, are enriched for astrocyte, microglia, oligodendrocyte precursor, and neuron genes, while PLS negative genes, which are more expressed in the blue components, are enriched for oligodendrocyte and endothelial cell genes. So overall, we conclude that a disordered thoughts, sense of self, psychosis spectrum dimension has a distinct signature in morphometric covariance patterns, that morphometric components converge with large-scale cognitive and functional architectures, providing a possible structural functional scaffold for PS emergence, and that components linked to disordered thoughts or sense of self may be coarsely separable from other components by expression levels of specific cell types. So thank you for listening, and I hope to see you at my poster.